name is Lucy, and today we're going to talk a little bit about how parasites have been shaping the human genome pretty much since the dawn of time, specifically focusing on the tiny little protozoa that causes malaria.
So, when we consider the genetic mutations that malaria has spurred along, we first have to understand what malaria is. Most people understand that malaria is spread by mosquito bites. But what might come as a surprise is that malaria is actually caused by a little group of parasitic protozoans called Plasmodium. These little guys often act as a parasite not only to their human host, but to the mosquitoes that carry them. The parasitic lifestyle involves a mosquito host infecting a human, then the parasites infecting the human bloodstream and being picked up by another mosquito. This means that Plasmodium spend a lot of time in red blood cells. When Plasmodium infect red blood cells, they reproduce in them and then explode the cell, not only releasing more Plasmodium into the bloodstream, but destroying the cell and causing many of the harmful symptoms of malaria, such as anemia, lack of nutrients, and fever. Basically, when too many red blood cells are destroyed, the body can't cope. So, how does this nasty disease spur genetic mutation? Enter sickle cell anemia, also known as sickle cell disease. As you can see, sickle cell disease causes red blood cells to be abnormally shaped, altering how they work and travel through the body. Now, I know some of you may be thinking, isn't sickle cell a bad thing? Well, yes and no. If you look at the maps in front of you, you can see that the one on the left demonstrates occurrence of malaria in Africa, and the one on the right shows the percentage of the population with the sickle cell allele, hemoglobin S. These maps show a strong correlation between areas where malaria is present and an abnormally high percentage of the population with sickle cell allele. Natural selection holds that advantageous mutations tend to stick around, while those that hurt people don't. This suggests that there is some benefit to sickle cell anemia, and in fact there is. Sickle cell anemia is the perfect example of the heterozygous advantage. The sickle cell allele occurs when one change is made to the HBB gene on the chromosome 11. It is an example of a single nucleotide polymorphism, and this mutation persisted due to malaria. While those who carry two copies of hemoglobin S tend to suffer from side effects of sickle cell disease and have a shortened lifespan, carriers of hemoglobin S, or those who only carry one of the alleles, actually benefit from resistance to malaria. The sickle shape of the red blood cell makes it harder for the Plasmodium to infect, and this reduces the side effects of malaria. While it is important to note that malaria never caused the hemoglobin S allele to appear, it did help select for its prevalence in the population by giving its carriers a distinct advantage. This fascinating example is one of thousands that might be something to keep in mind the next time you think about your genes. Thanks for watching!